Hello and welcome to this evening's UCSF Alumni event, Alumni in Film, Human Nature. Thank you for tuning in. I'm Mario Praza, Executive Director of Alumni Relations. Before we start tonight's program, I want to review a few housekeeping items to help you with your experience. Just so you know, we are recording tonight's program and we will share it with you via email in a few days. Tonight's panel will be driven by your questions, so I definitely highly encourage you to submit any and all questions you have about the film and Crispr in general. To submit a question at any time, please use the Q&A box at the bottom of your screen. Please be sure to keep your questions as brief as possible. Now, please join me in welcoming the host and moderator for tonight's program, the Executive Director of Human Nature and Executive Producer of iBiology and the Wonder Collaborative, Elliot Kirschner.
Thank you, Mario, and thank you all of you for joining us. We're looking forward to talking CRISPR tonight from many points of view. And as Mario mentioned, my job is to really more moderate than generate all the questions. Really would love to hear what you're thinking and the questions you want to ask. So please put those in the Q&A section. We'll sort of get to them throughout the evening. I want to bring the other panelists on now, and we're going to do sort of self-introductions once they pop up. So maybe as you pop up, Sarah, first is Sarah Goodwin. Would you want to introduce yourself, Sarah?
Yeah, hi, I'm Sarah Goodwin. I'm the Executive Director of the Science Communication Lab, which is a small nonprofit dedicated to telling the stories of science. And it includes iBiology and the Wonder Collaborative, which is the entity that produced this film. And I was the lead science advisor and a producer in Human Nature. And I'm an alumni of UCSF. I got my PhD in cell biology in 2011.
Okay, Navneet, if we want to turn your camera on and introduce yourself as well.
Hello, I'm Navneet Matharu, Assistant Adjunct Professor at UCSF, Principal Investigator at Innovative Genomic Institute. And I first heard about CRISPR in 2012 and with the Jennifer Doudna's article in Science. And it was actually kind of amazing to see that you could create mutations in any organism if you want. Being a geneticist, I was pretty excited with the ease of doing the system. And 7 years down the line, I have like first-hand experience of working with the CRISPR system. And here I am for the discussion panels.
Thank you very much. And Ethan, thank you for joining us.
Yeah, I'm Ethan Weiss. I'm a physician scientist, cardiologist, Division of Cardiology, and also Cardiovascular Research Institute. I came to UCSF in 1998, so 23 some odd years ago, to do my cardiology and research training, and I've been here ever since. And I am here tonight because I had a very tiny little bit role. Really, my daughter, my now 14-year-old daughter, was somewhat featured in the film.
More than somewhat featured, and I mean it's and actually I think that was one of our first shoots. So you were both a subject and a guinea pig for our efforts in making this film, which was a long and winding road, as you know. But I think we're going to dive into the film. But I think hopefully we can talk about both the film and CRISPR and and how they came together. But Navneet, you started a little bit with this, so maybe we'll come back to you to finish up. But I'd like to ask the other two about when you first heard about CRISPR and then the journey since then, including maybe the film, since you're both were involved with the film. So maybe Sarah first, if you when did you first heard about CRISPR and what transpired since you've heard of it?
Yeah, I graduated in 2011, and Jennifer Doudna and Emmanuel Charpentier's paper came out about a year later in 2012. And, I was still in touch with a lot of colleagues. So at that time I had gone to iBiology already and was working kind of in science communication, but I still had a lot of friends who are still in the lab, both in industry and in academia. And I just started hearing about CRISPR being used as a tool by a lot of people. And it just kept increasing and increasing and increasing. Until we were just like, all right, I really want to understand what CRISPR is, why it's so exciting. And I had actually been a fan of Jennifer Doudna's work in Dicer. She'd done other work in RNA systems before that I had really been fascinated by before I even joined graduate school. So, you know, she was a leader in the CRISPR work. And so we invited her to give a talk for iBiology. I think it was in 2015. And she gave, there was a green screen kind of a lecture talk about CRISPR and how you could use it. But at that time, it was clear that CRISPR was just being used all over the place in the labs, and that it had a lot of potential for really impacting medicine and non-human uses as well. And so we did another, so Elliot, who is a part of the Science Communication Lab, has worked with Dan Rather for a long time. And we decided to have Dan interview Jennifer Doudna. And I think that was about a year later after her talk. And they had this great conversation. And a part of that conversation was Jennifer kind of toiling with the fact that, you know, there's this powerful technology, CRISPR, and just like she was talking about going to a PTA meeting at her school and just like no one knowing about it. And what kind of transpired from there was the idea of doing a video about CRISPR or a short series about CRISPR. We weren't really sure what it was going to be, but we gathered a bunch of people, including Jennifer Doudna and other people in the film, for kind of a summit on CRISPR, and decided that it really deserved a documentary film where we could talk about the discovery that was in basic research, talk about the applications, and really dive into more of the societal and ethical issues. And that was kind of the genesis of the film. So I was involved just through my involvement at iBiology, but it was a really interesting journey and experience.
Ethan, you know, I mean, we talk about actually in the film sort of when you first sort of heard about it, and it's in, you know, heard about it from a scientific point of view, and then its sort of impact and thinking about genetic engineering and all of that, you know, from your personal story. But maybe if you could drill down, when did you first sort of hear about it, and how did it amplify as time went on and merge with sort of your own story?
Well, I mean, so I distinctly remember the first time I heard about it, I was at a cocktail party at a faculty member's house, and Jonathan Weissman was talking about this new technology called CRISPR. And, you know, whenever Jonathan talks, I listen. So I listened, and he described it. It sounded pretty amazing. Now, I mean, there were other gene editing tools back then, but this one sounded different. And, you know, that excitement continued. I think, you know, sometimes in medicine or in science, we get excited about the new bright, shiny objects, and then you stop hearing about it. But this one just never went away. And it was, you know, all over the place, and eventually sort of penetrated, I think, beyond the sort of scientific community, and it was all over social media. And obviously in the film, I talk about, you know, I had this exchange on Twitter with a scientist around the sort of potential uses. He had said something about using CRISPR basically to like a vaccine with kids with monogenic diseases. And that resonated for me because I've got a kid with a monogenic disease. So that sort of really started the process of sort of merging the science with the personal. And then Erica Check-Hyden, who's an amazing writer, wrote a feature about my family, about Ruthie, my daughter, in Nature. And I think Adam or one of you guys read that. And Adam reached out and asked if we'd be willing to be a part of the project. And we said, sure. Well, it wasn't quite.
Them being Adam Bolt, the director, who did obviously a wonderful job. It's our bias, but we think he did a great job in telling the story. And, you know, he himself doesn't have a formal science background, but just really brings a lot of intelligence and curiosity to it. I mean, I'm curious, Navneet, you know, as we, as Ethan mentioned, you know, there are a lot of new technologies that emerge that seem big or, you know, sometimes get overhyped. You know, CRISPR, one thing that struck us as sort of storytellers was that, you know, oftentimes you hear something and then you go to the scientist and it's, yeah, don't get too excited, or it's, you know, it's not as big as it seems. This is sort of, it's overhyped. But this was one of the cases where the scientists seem to be hyping it more than anything. You know, that seems like this is really that big. This is really that transformative. When did you get that sense, you know, in your own research and story about how big this was? And did you even early on, did you have a sense of where we would be now, or are you surprised by how big it is, or did you think it would be bigger maybe?
Yeah, I think being a geneticist, the forte for any geneticist would be actually to change the nucleotide sequences at will, for especially in organisms, let's say in mice or, you know, in flies, because that's how actually you learn genetics. And there were other technologies that Ethan mentioned that starting from random mutagenesis, you can sort of change things in the genome, but you don't have control over. But then there were other technologies that came in, like zinc finger nucleases that could actually change the sequences that you want. And then came the talons, which is the plant-derived methods, the binding domains that could actually, if you stick a nuclease cutting domain with it, you can actually create mutations. But actually CRISPR was, what struck me was the ease and the adaptability of this system for researchers worldwide. Anybody actually can, have a Cas9 CRISPR system, design a guide, and then target a favorite position in the genome. That was actually a big sort of a breakthrough, that the adaptability and the ease of use of this system was far reaching. The reason is, I was actually in 2012, I was discussing that, hey, I want to create, I want to do this genetic mutation in zebrafish, and somebody told, oh, CRISPR has the answer, you should use CRISPR. And any genetic tool, such genetic tool has the therapeutic value also. Zinc fingers has been used for, have been used for actually therapeutic purposes. But the ease of doing this, and you can actually target any sort of location with extreme specificity, was just so much powerful for CRISPR system that was not applicable for zinc fingers and talons. And that was the time I thought that something, if you can create mutation, that might have actually a huge and long impact in rescuing or creating a therapies for this mutation. So that inspired me a lot. And what I actually started doing is started working on non-editing versions of CRISPR, and see if we can explore the therapeutic potential of non-editing versions. And as I said, that it just goes on and on and on. The CRISPR system, the expandability, the toolkit it's so diverse that maybe it might take another few decades to understand the full potential of CRISPR system itself.
I think one of the things about it that we even saw over the course of making this film is how quickly this is developing, and that that we're already in places we couldn't predict. And just maybe for just a little clarification, when you say non-editing, what would be a quick explanation? What do you mean by that?
Yeah, for the general layman audience, CRISPR means you can actually cut a DNA or edit, which means that CRISPR has those cutting scissors. But if you actually deactivate those scissors such that it cannot make a cut now, but it can still target the favorite location in the genome, you can modify CRISPR to do whatever you want other than cutting the genome. So what are the other ways you can do it? The toolkit is actually even bigger than the cutting CRISPR itself. So I can definitely see the impact and influence of this CRISPR system in developing other adaptable versions and diverse version of CRISPR to be really far reaching.
Thank you. I'm going to jump to one of the questions from the audience, and please use the Q&A section to keep them coming. This is from Melissa Hendershot, who says she loved the film, which we are pleased with. She loved that the film included some perspectives and medical applications that I wasn't familiar with. How did you find these stories and decide which stories to include in the film? So I'm going to maybe address that, Sarah, to you first, and then maybe Ethan to follow up on what it was like being on the receiving end of a call to be in the film, and what you sort of felt about that, how that process, and I think that could lead to another question. But how did we decide what stories to tell, Sarah?
Well, it was really important to us, I guess, first and foremost, to feature the discovery story of CRISPR. So kind of almost ending with Jennifer Doudna and Emmanuel Charpentier's paper, and starting somewhere else, because it was such a beautiful basic research story that we just felt like would be interesting to be told, because it included, scientists who lived internationally. It included people who worked in industry. It's just a really fascinating story. And so, to find those stories, that's actually where I played a role. So I was kind of the scientist on the film team, and I just ended up reading a lot of papers and diving into kind of the story myself through the primary literature, which was actually a really fun thing for me to be able to do. And so we decided who to interview kind of based on the papers that came out and the pieces that we felt like would be important to tell, to kind of give an understanding of CRISPR through the discovery story. And, you know, to be honest, that was kind of a risk we took. You know, a lot of, we just felt like stories about basic research that really dived into the process of science were missing from kind of the public consumption. And, you know, we heard from funders, some funders who didn't fund us, but a lot of funders did fund us and really took, you know, a leap of faith that we could tell a compelling story. But we heard from others saying like, that's just not, you know, interesting or exciting. But we really were committed to doing that. So that was kind of, you know, that part of the film. And that was really based on who was doing the research. Although, you know, we lucked out in some ways with some of the people like Rodolphe Berengu, who's such a fantastic character as well, in addition to his research, and other people. Like, you really got a sense of the personalities of all the different scientists, which I think was really, you know, something that was really special and fantastic about how it turned out. And then, you know, there were other people, like we really needed someone who could kind of connect the dots in a lot of ways. This is still on kind of the science side. And so we found Fyodor Urnov through, you know, a colleague of Elliot's father, who's a scientist, and that was kind of lucky. But he's at Berkeley. He's a, you know, a major player in the gene editing field, and he just is wonderful explainer. He's an educator too, and you know, he's just was a huge asset to the film. And then you kind of go to the other side, kind of the non-science stories, and that was, you know, there's a little bit of serendipity in terms of, you know, coming upon the article that Ethan mentioned. David Sanchez, who's in the film, also was someone, you know, we called around to different scientists who were doing sickle cell trials and just seeing if, you know, there were any patients they worked with in sickle cell that they'd connect us with, because we really wanted to show, you know, the side of a patient story who had a really kind of, you know, debilitating, life-threatening genetic disease. I mean, you know, of course, there are ones that are even more severe than sickle cell, but sickle cell we felt like was a good example of, you know, a genetic disease that we've known the genetic basis for a long time, but just still haven't had a treatment for. And, you know, of course, CRISPR is being used for that now. You know, and another big character I'll just mention is Alta Charo, who was the chair, one of the chairs of the National Academy of Sciences meeting about CRISPR. And we really used that meeting as another kind of point of how the scientists and others were being involved in the ethical and societal discussions around gene editing. And this National Academies report was such a major player at that point. And so that was also good. You know, there are a lot of stories that didn't make into it. I'll just finish with that thought. Is there are a lot of things that didn't make it into the film. We had a lot of more content on stories about CRISPR being used and not for human purposes. Like we had a whole thing on coral that didn't make it. We had a lot more on the earlier gene editing technologies and some great interviews with like Paul Berg that didn't make the film. You know, hopefully we'll be able to use it in other ways, but that was a hard process. So yeah, so I guess a little bit of just kind of the story of CRISPR, a little bit of serendipity, and then, you know, the willingness of people like Ethan to, you know, to share their stories on camera and let us kind of invade their families for many, many days, which we really appreciated.
So that leads to my next question. I mean, Ethan, you know, you get a call to be in a film like this, not knowing, we didn't have any track record as, you know, we were a new organization and all of that. You know, what made you and you know, especially Ruthie and your family, you know, agree to do this? Why do you think it was important? And how was the process from your side of sort of telling your story through a medium like film and seeing how it, you know, I mean, in some ways you're you know, very trusting. You know, you gave us whole movies and other things to, you know, use to tell your story. And why do you think it's important to tell these kinds of stories? And how did you know, how was it like from your side of the process?
Well, I'll answer that one last. But look, I mean, we had this kid in 2006, and she has this genetic condition. For those of you who haven't seen the film, it's oculocutaneous albinism. Colloquially, she's called an albino, fair skin. And what I didn't know until I had my own child with this condition is that one of the most sort of important features of this condition is a very severe impairment in visual acuity. So she's legally blind, and we were devastated when she was born, or when she was diagnosed around 6 weeks. And there's no doubt in my mind, and I don't think we got into this in the film, but I've written about it elsewhere. There's no doubt in my mind that had we had the opportunity to edit her or fix her or even not have her at that time, we would have taken it. But of course we didn't have that. And so we had this kid grow up in front of our eyes, and she turns out to be pretty special. I'm biased, right? I mean, I'm her dad. I think both my kids are special, but this one is pretty special. And it was pretty cool to watch the way she impacted the world around her as much as it impacted her. And I thought it really, you know, we talk about diversity a lot, but watching her peers exist in a classroom environment with a kid who can't see very well, and learning how to advocate for her, and her learning how to advocate for herself, and all these things, it just felt like this is a part of our existence on this planet that it would kind of be sad if it didn't exist. And so when I read this, you know, tweet, it sort of triggered me a little bit to kind of think about what would have happened if we had this technology. And the very first thing I did, and I don't want to go on for too long, but the very first thing I did was to go to talk to Ruthie about it. Ruthie at that time was in 4th grade, and she was like 9. And I asked her if she had the opportunity to kind of make this condition go away, if she would. And she said no. And anyway, this all turned into this article that you know, introduced us to you. As to, you know, why we said yes, maybe we were dumb. We actually had some friends who were documentarians who told us that we should not do it. I think we talked about it a lot as a family, the 3 of us. So Ruthie and Palmer, my wife, and I talked about it. And we felt like the main reason to do it and to sort of put ourselves out there and take a little bit of risk was that there are kids born all the time with these kinds of conditions, and parents are left feeling like it's the end of the world. And we wanted people to understand the perspective that we had, which was that this kid was not the end of the world, that she was quite the opposite for us. And so we felt very fortunate to have Ruthie not just exist in this world, but to exist as she did with this genetic flaw that didn't get fixed. And so that was sort of the perspective. The experience was amazing. I will say, to follow up on what Sarah said about cutting, I mean, you guys probably spent 3 weeks with us on and off, and including a trip to Tahoe. We all went up to go skiing. And I think we spent like 3 days up there and had all these cameras all over the mountain. And not 1 second of that ended up in the film, which wasn't a bummer for us necessarily, but I just couldn't imagine how much money you guys must have spent on things that, like, because that was probably not cheap, right? Getting a whole.
No, I gotta say, we should get you some. It's absolutely beautiful footage. I mean, we should find a way to get it.
I saw some of it actually, because it was in an original cut that you see.
Yeah, it was. Yeah,
and it was beautiful. And I thought, there's no way they're not going to use this.
I know. It's just, I mean, it's, yeah, there was so much. I mean, I think part of the issues about this is that the story, you know, one of the exciting parts about being a storyteller, but the struggles is what do you put in and what do you take out? And you know, it's just, you know, we had a real decision, you know, to try to make this, you know, and we wanted to make it a film because we wanted something people could sit and watch together in one sitting, as opposed to a series, which is over time. And, you know, that made some really hard decisions. I want to jump, there's a lot of questions about the science, but there is a question from the audience here that I think is a good follow-up for you, Ethan, and maybe others can discuss. And that it's brought up in the film and you've alluded to here. Would you discuss the issue of consent? What does it mean for a parent to make a decision like this for an embryo or infant about its genes? Are there other situations in which one's genes might someday be edited without their consent, i.e. people convicted of violent crimes or something like that? I mean, I think this is one of the real questions that comes up in both in Ruthie's story and then in David Sanchez's story too, the other young patient that with sickle cell, about, you know, would you change it? Whose decision is it to change it? And this is a change, you know, the nature of, especially if you're editing in the germline, you're not just changing an individual's genetic makeup, you're changing their progenies by, so how do you sort of see that issue of consent in terms of your own story?
Well, it's fascinating. B, I think it's really fascinating that both Ruthie and David both said that they wouldn't want to change themselves. Now, I've heard people tell me about Ruthie that she will eventually want to change herself as she gets older and she can't drive a car or something happens, you know, to her socially, but she's been pretty consistent in her own opinion on this throughout her, you know, sort of young adult life. The consent thing is really interesting, right? So there's 2 issues in the way I look at this. One is if there's a kid or a young adult, then then you have a question of, can they give consent? How does that work with a parent? But the more fascinating thing for me was how if somebody had approached us as parents in 2006 when Ruthie was a baby about doing this, how do you consent for something where you don't know what the outcome is going to be? So in other words, like, it would have been very difficult to say no because we didn't know what Ruthie was going to turn into. And so I think, you know, it's almost unconsentable because you can't know what the experience is going to be until you've actually had it. And so it is a very difficult problem. Now, there are many conditions where kids suffer and have diseases that are tremendous burdens on them and their families. And I don't think there's as much or any question about what people would do to try and help these kids. But in Ruthie's case, I think there really was a conversation. And again, we would have consented to do it, but we would today regret having done that. So.
Yeah, it's sort of, I think one of the purposes, I mean, one of the real places that her story plays in the film is it pushes up against, I mean, it's easy to say, okay, yeah, we're not going to do it just for hair color, or, you know, and of course for, you know, Huntington's disease, we're going to do it. But as you start, you know, getting into what makes diversity, where it's not a fatal disease or a painful disease, you know, it's sort of, what do people opt for, and, you know, where's that gray line?
Well, and Ruthie's, I mean, again, this comes up. Ruthie's condition, OCA2, is on a bunch of pre-implantation genetic screening panels, and so there are, you know, probably a lot of parents out there who see they have this or that they're each carrying a allele, and that they are deciding to not have a kid with albinism.
Yeah. Well, I want to move on to some of the science here. And I mean, this is a question I don't know whether you feel happy to answer or go for it, but one of the questions is, you know, and some of the regulation around this, you know, about whether, even especially for non-human applications, about whether CRISPR is genetic engineering or not, or GMO. You know, these are big questions for how it's regulated, and there's some disagreement about, because it's so seamless and it can change. It's not necessarily taking DNA from a foreign or from a different species and dropping it in. But the question is there a lab test which could detect when someone's genes or germline have been edited? And maybe I would just sort of, you know, say, by extension, maybe not just if you consider, like, a lot of people expect that the applications are going to appear much more early in agriculture and in other types of situations rather than humans. You know, can you go and say, wow, this genome has been edited because of CRISPR? Does it leave any evidence of that?
I think that's kind of a difficult question. If you actually do a somatic editing, you know, let's say after the person is born, post-natal editing, possibly you can tell. Let's say, you know, for example, I edited, you know, some of the regions in the eye, you know, some of the cells in the eye. And at the same time, the other cells in our body would not be replicating, you know, exact matches what CRISPR editing has done. And if you think about the germline editing, you know, if an organism is edited at the germline level, I think the way CRISPR makes its edits, it create indels, it leaves certain portions, deletes, or, you know, includes certain portions in the genome. We can always go back and look at the frequency of that mutation in the general population and compare it, that the frequency of this edit, or, you know, this particular mutation is not found in any other second individual, which actually could suggest us that it could be edited, but there is not definitive proof that, you know, whether it was edited through CRISPR or it was edited through some other sort of gene editing tool. So I think it's a good sort of genetic problem, which you can probably tell that, you know, this has been edited at a sequence level.
And this is sort of a follow-up. I mean, presuming that is that another question from the audience? Will it be possible to conduct safe, ethical trials of gene editing in humans? Now, we already are seeing that, right? And are those different from other forms of clinical trials? Is there certain extra concerns? I mean, how do those work?
Yes.
And what's the state of some of these trials?
Yeah, so there are CRISPR trials undergoing for certain conditions. One of is the retinal disease in the eye, liver disease, or sickle cell disease, as your movie also, showcased. Uh, there are strict and separate rules and regulations provided and guidelines provided by FDA for setting up those clinical trials as well, which are different from any other pharmacological modality. Some of them guidelines will be overlapping, but FDA is cognizant, is aware of the fact, what is the impact, what are the side effects, what are the toxicity of the CRISPR system itself. And the clinical trials are designed specially with the FDA guidelines for the CRISPR system itself. So the rules of the CRISPR trials for a game are pretty much set, and FDA actually make sure they are followed well.
Now, Ethan, I mean, one of the things that struck us about CRISPR is its ubiquity. I mean, it's a tool as opposed to, it's a narrow discipline. I mean, your own background in medicine and your own research. I mean, it's not that you came up with genetic engineering as your specialty, but yet, how do you see the echoes of what CRISPR is already doing in medicine? I mean, how much are we seeing with Lab Bench? Not in this whole thing is pretty recent. How are you seeing it in the clinical side, and how varied across medical disciplines are we seeing its impact?
Well, I mean, I think gene therapy has been around as, for 25 years around. And so we've all been thinking about this. I think there are a few things that are different with CRISPR. One is the sort of ability to legitimately edit in vivo, whether you're editing somatic cells or germline cells. And I think where things have gotten very interesting is sort of that discussion around editing germ cells. I don't know of anyone who's doing trials in editing germ cells. Navneet, you may know, but I think for all intents and purposes, that's still sort of off the table from an ethical standpoint. But there are lots of trials, using CRISPR and other forms of gene therapy. I mean, anyone who's gotten a vaccine from Pfizer or Moderna has had gene therapy effectively. And CRISPR is used in a lot of there are many assays to test for COVID-19 that employ CRISPR technology. So we've seen this sort of explode in the past couple of years, both, I think, broadly speaking, gene therapy, where there are a number of approved therapies now, and then, more recently, specifically to CRISPR.
But a follow-up specifically, I mean, obviously it's a disease a lot of people care about. I mean, it's not just one disease. But I don't know if I'm pronouncing the name right, but Michaela Presti asks, how is CRISPR being used in cancer research? Anyone close to a clinical trial? There are applications, for this in cancer. Does anybody want to jump in on that?
I mean, some names come to my mind if you heard about the CAR T cell system, hematopoietic cancer cells or leukemia. I think there are certain sort of, I mean, based on published research and industrial efforts that actually are aligning towards cancer therapeutics as well. So maybe in next few years, we'll see fruits of this labor. But I think it's a sort of very open field, actually. It's an amenable field for CRISPR, yep.
And I mean, both to Ethan and Navneet, do you think that the pace that we're going to just see a growth in the amount of therapeutics that are derived from this, from using CRISPR? I mean, do you think that there's some targets that are successful days like that, or do you think that it's just going to grow in relatively soon in terms of its impact on medicines going into clinical trials?
I think that the place where you'll see it the most are in the diseases involving cells that you can take out of the body. I think that's, you know, like sickle cell, right? I mean, one of the advantages of sickle cell or blood-based cancers is you can take the cells out, you can edit the cells and then put them back in. There are people obviously doing trials or starting to do trials to try to edit organs or cells in vivo to actually, the hard part there is delivery. It's something, fortunately, that people have been thinking about a lot with other gene therapies. So I think, you know, again, I think this year, the past couple of years we've seen explosive growth in gene therapy in general. And, you know, CRISPR is still relatively young, but I think CRISPR-based gene therapy will explode. I mean, we're seeing a tremendous, this past couple of years is really seeing a change in the way that we deliver drugs to people in a way that we really haven't seen since the advent of monoclonal antibodies. And you know, before that, recombinant proteins. I think we're seeing so many different kinds of new technologies to basically use as a drug. And that's really exciting.
Yeah, I agree with Ethan on that. CRISPR, when actually in human nature also, you talked about the SCD, the sickle cell disease, which is the very good example of ex vivo. Ex vivo means you take out the cells out from the body, modify it, and, you know, put them back in. But as Ethan mentioned, that in vivo, you know, in vivo, in somatic CRISPR deliveries, there are certain companies and clinical trials that we might actually see in, you know, very near future. As you know, eye is actually a target, liver is a target, muscle is also a target, like skeletal muscle for DMD, you know, patients with muscular dystrophies. So I think I think we will end up seeing a lot of development happening at the therapeutic end of the CRISPR in next sort of few years or so. And both in vivo and ex vivo would be actually figured out when you talk about the delivery issue. I think I'm pretty hopeful and pretty optimistic.
Great. Well, we're gonna shift now back to talking about the film. And Sarah, this is a question directed to you from the audience. I don't know the person's name, but as a fellow alum, I just want to say how very cool you got to serve as the science guru, in addition to being a producer for the film. Can you share what working on film has been like for you and what advice you have for any of us considering doing something like this?
Yeah, it was a really amazing experience, being able to use my science background in this way. It was kind of invigorating getting to read papers, and there are a lot of sequences in the film, and I looked all of those up and figured out what mutations needed to be highlighted and to make sure they were scientifically accurate. But kind of beyond the science, I learned a lot about storytelling through this process. For me, it's just like, yeah, of course, CRISPR is interesting. Everyone should know about it. And the discovery story is fascinating, but how do you actually make that into a fascinating story for a lay audience? And that's something that we spent a lot of time thinking through and talking about. I mean, some of the decisions and what we included and what we didn't were really based on what we thought the audience's journey would be for people who had never even maybe heard of CRISPR before. And really taking them on the journey of, not only how the science works and how it was discovered, but also just giving them a lot of different, things to think about. I think, something that we really felt strongly about in the film was that we didn't want to tell people what to think. We wanted to kind of show a bunch of different perspectives. And all of this was stuff that I never really appreciated this about film before working on this. This is the biggest project I've ever worked on outside of my PhD work. And so I just got a much better appreciation for how to tell a story from this. And, I think that for people who are interested in pursuing this kind of realm of science communication, something I've learned, since working on human nature is just the lack of scientists who work on science films like this. There are exceptions, like, HHMI has a lot of scientists who work on their films that they put out. But, I've been to a lot of conferences, and a lot of the conversation is around, like, in human nature, like, we had all these scientists who spent hours of their time, like the interviews are hours long, that we had with these really, amazing scientists talking, about their science and just their thoughts. And when we go to these conferences, they're always like, how do you get these scientists to talk to you? Like, how do you get them to spend any time with you? And I think a big, part of that is having, scientists on the team, on the production side, that can speak the lingo of scientists, that, can give scientists the confidence that you're going to tell their story right. And you're not going to, we really made it clear we weren't going to dumb it down. We were going to really give the nuance of CRISPR kind of the time it deserved. And I think having a scientist who was working with the film team gave some of that, confidence in that. And, someone who could understand kind of the culture of science and how science works and why it's important to include that in the science stories. And so, there are a lot of different ways to get involved in science communication efforts. And, you can email me and I'd be happy to talk about them offline. But I just think that there really is a big role that scientists who are interested in communicating to the public can play in all sorts of science media. I mean, we've even seen with the coronavirus kind of what, kind of a mess it's been communicating science. I mean, that has its own challenges where it's just the pace of it and the unknowns and the uncertainty. But I think that, people have really struggled to find clear information, for a multitude of reasons we don't have to go in on this call. But, I think the role of the science turned communicator is a big one that will just become increasingly important.
Yeah, I mean, I think it's really important that, that scientists really become part of the communication of this, and that, part of what we're trying to do at iBiology, and, we're sort of under the new umbrella of what we're calling the Science Communication Lab, is really bring storytellers and scientists together so that storytellers can learn about the science and the scientists can learn about the stories. You mentioned, again, Sarah, that we, and there's all this material that we didn't make the film, and some people in the comments have wondered about us publishing it in other forms or releasing it in other ways. I mean, a lot of that is, dependent. I mean, a lot of it's in a very raw form, and so it's dependent on budget and time to edit these into something. But, and I think that is something we're really interested in doing, is taking some of the materials. And we did actually do that. We should say that it's available online. We had very long interviews with Jennifer Doudna and Emmanuel Charpentier, as well as Martin Janik, who also worked on that major experiment, cut that together into a story about their experiment, where it's much more in depth for that groundbreaking experiment, which led to the Nobel Prize. And so we are going to be looking for opportunity to re-edit this stuff or put into new formats. So yeah, I'm going to switch back. One of the things that there was a real focus on human, it's on human use for this technology. The film is entitled Human Nature, but we do allude to its use in non-human life forms. But here's a question about that, really, which is the question of gene drives. So I'm going to throw out the question, and I'll leave it to others to better define what a gene drive is in case there's some confusion, and also sort of talk about what, this is something I think that does scare a lot of people, the notion of a gene drive. So the question is, a gene drive conjures fears similar to a nuclear weapon falling into the wrong hands, other than the currently being rather difficult to do. And the question is, how difficult is it to do? What safeguards are there that this doesn't get out of control? I mean, for those of you doing the research, maybe, I don't know, Navneet, if you want to talk, I mean, you probably don't think you work in gene drives per se, but this question of its use, what it is. And then I'm curious, both, for you and for Ethan as well, like what kind of regulations do you see around this from a practitioner point of view? Are people using this technology or knowing of it?
I think we talked a lot about, the CRISPR and its beneficial effect as a therapeutics. We can sort of help humankind to alleviate certain diseases, which is, I think everybody sees it as a good thing. When it comes to a question like gene drives, or questions like, some of the so-called unethical and ethical gray areas of use of this technology, whether you want to use this technology for some of the what you call like a trait selection, or some sort of characteristic selection in a human population, which doesn't have a direct therapeutic advantage. I think for those discussions, I want to actually also point out that it's always important to see a technology with the benefits and risk. So if you think about that, changing an eye color would be something, a beneficial trait, but the risk associated with changing the eye color for CRISPR could be large, which actually the regulatory authorities would not like that, because you want to actually balance risk versus benefit ratio before putting in such a profound technology into purpose or into application. So my understanding is risk versus benefit ratio would be really high for these kind of applications. And I think the regulatory authorities are aware of these and would place in correct sort of required regulation to whether it's ethically to move it forward or not. As in movie also, Charo actually really discussed this issue of bioethics and really well. Ethan, you want to add something?
I mean, I can't really speak to gene. I mean, look, there are potential uses that are good. There are potential uses that are, who knows, the unknown unknowns, so to speak, to kind of borrow from Don Rumsfeld. And then of course, there's the potential for bad, right? I mean, when COVID came, there were lots of conspiracy theories that this was engineered in a lab. And to my knowledge, those are all false. But I think any of us who understand the science would know that doing something like this is not impossible. I mean, maybe it would be hard to take an existing coronavirus and make it into what we got, but we certainly, people, if they wanted to, could make it worse, right? I mean, there's a blueprint to do that now. And yeah, so I mean, there are potential things that could go wrong. Anytime we introduce new technologies, that can happen. It happened with nuclear weapons, right? I mean, we know that there are bad people out there who are going to do bad things.
But as far as I think the regulations are in place, and Ethan, I think you agree also, like we as researchers or as scientists, we are also tightly regulated in what sort of activities we can do. There are actually IACUC protocols and the guidelines set for researchers to do. But at the end, I think if somebody wants to use it not for the good, but for the bad of the mankind, then I think I'm yet to see if there are certain organizations and countries that can actually sort of control those sort of activities.
The one thing I'll add to that, just in gene drives, is there's a scientist at, I believe at MIT or the Broad, Kevin Esfelt, who has done work on gene drives and has spent a lot of time talking to the community about, so this is specifically like Martha's Vineyard or Nantucket, where ticks and Lyme disease is a really big problem, and could use gene drives to eliminate that. And and he's gone on to talk to other communities about this, and it's interesting. He's really taken the point of view or something like gene drives, where it really has this a snowball effect, that you need to have community consensus to introduce it. And I think that has sparked some really interesting conversations he's had with community members. It's not in our film, but I think he's talked about it in other venues. And it really kind of brings up some of these questions about who are the deciders with certain technologies.
And I think that was part of our hope in making this film, is that, while they're the scientists, the researchers that are doing this, that these are questions about use, and people don't understand necessarily how all technology works, but that doesn't mean that they can't be part of the discussion about how it's used. And the hope is that more people will feel comfortable to join that discussion. We have about 10 more minutes left, so please keep your questions coming in the Q&A section. One thing I did want to ask each of you, though, is what has surprised you about CRISPR at this point? Is there and sort of where we are, or maybe if you want to take the question a slightly different way, what back to the known unknowns. What might surprise you trying to predict the future here, looking forward around this technology?
I could start maybe from a bit of the non-scientific, well, I guess it's a science view too, but so our film wrapped up right before or right around when JK, the scientist in China, had announced that he had edited two born girls, which that was shocking to me. I just thought that something like that was much further down the line. Now, that came with a lot of backlash and condemnation, and, that we're not ready for this. And I think it's discussed, it's, brought a lot of dialogue, and now there's these two girls who exist with these gene mutations made by CRISPR edits. There's questions around those. I mean, that really surprised me. And, I don't know what's going to come of that, but that I'd say that's just as a moment really struck me as something that came a lot sooner than I expected. I think these questions around consent also are really interesting. I had two kids, I had one child while we were making the film and another soon after. And just kind of these questions that Ethan grappled with were really fresh in my mind as we were talking to different, to Ethan's family, talking to genetic counselors, just like all of those things that we had going on around the film while I was also, thinking about my own kid and, the decisions that I would hypothetically make if confronted with some of these decisions or issues. And I think the last thing is just the thing that we don't really cover as much in the film is the use of CRISPR in non-human cases. And, most people we talk to say that that's really, it's already happening, like, CRISPR being used in different agriculture examples and just how it's going to really come to consumers in that way. And so I think that's where I'm curious to see, what the next 5 years brings and also kind of how that is received by the general population.
Anybody else want to jump in? Ethan, what's surprised you about this? Is there any, or what are your thoughts might surprise you going forward?
Well, I guess the most surprising thing is that this was discovered 8 years ago. And here we are talking about human clinical trials and all these incredible applications in and out of human beings. And we've talked about incredible potential upside and some downsides. We're just moving very fast. And I guess that makes me always a little bit nervous when we move this fast. Obviously, there are a lot of problems we need to solve and should solve. And people are excited about this technology, but it occurred to me throughout this whole process that it's very hard to put toothpaste back in the tube. And so I hope we can all take a deep breath every once in a while and think about what we're doing.
I think what's interesting about this, and I think that's something we try to sort of lay out in the film, is that you say that genetic engineering existed before this. A lot of what was needed to be done about this, about sequencing and a lot of other technologies were already in existence. That this was sort of a perfect way to sort of target this. And I'm sort of struck by this question of speed. And, one of the things that I think for a lot of the public, there's sort of an uncertainty around speed. It sometimes takes a while. We've had a quote unquote war on cancer for generations now. And, where sort of people feel like, oh, how are we doing? On the other hand, sometimes, all this basic research is just laying the groundwork and the right spark, hits it. And when you talk about speed, I'm thinking about how quickly we developed really very, very effective vaccines for COVID, which is, incredibly, I don't think anybody really expected that they would, whether we could distribute them and all, that's all these other social problems, but the actual science around how effective they are, how safe they are, how quickly they were developed, and multiple ones too, not just one, is quite, so I'm just curious, a little bit of question about the basic research that exists around this, that, in some ways, there's all this knowledge and then there's something new that just clicks at the right time. And is that really where we are with CRISPR? And are there other things like that are looming?
I think that also kind of gel well with your previous question of surprise, the surprise of CRISPR. As a geneticist, I know there are a lot of monogenic disorders. There are actually, there are a lot of rare diseases, more than 7,000 of rare diseases. There are a lot of orphan diseases where orphan diseases mean nobody actually developed therapies for orphan diseases. Like no pharmacological modalities, nothing. For those kids, there are no other options, because nobody works on that. I think what will gonna surprise me if actually CRISPR can, going by the safety and toxicity data, and if you check all those boxes for CRISPR, what will surprise me is CRISPR can actually bring an answer to give a therapeutic, answer to many of the monogenetic disorders and many of these rare diseases, which are nobody is actually thinking about. So I think I'll be pleasantly surprised in next, maybe a decade or so, that the speed with which we are going, there could be hundreds of those disorders that CRISPR can actually bring an answer for.
Well, thank you. We only have a couple more minutes. I want to thank everybody for your wonderful questions from the audience and all the panelists, and just be able to have this forum to discuss this here. It's just been wonderful. So as a sort of final question before I throw it back to Mario, just quickly, we sort of started with bio-optimist and bio-pessimist questions, or sort of this questions of where do we come down? Are you each of you more hopeful for the future around this, or fearful? We have about a minute left, so maybe Navneet, you hit this a little bit, and hopeful. And Sarah?
Absolutely hopeful.
Yeah, I'm a bio-optimist, I think, about a lot of things, so definitely CRISPR falls into that too.
And Ethan?
Both. I'm the old guy, and I'm I see the potential, but I also, I also see the potential for how this could come off the rails. So,
and just sort of, I don't want to leave on a pessimistic note, but what would that look like briefly, like it off the rails? What is the? I mean, there's often like really fantastical, considerations, but realistically, what do you think a scary application might be?
I'm not talking about like somebody intentionally trying to harm the world, or even somebody like, was portrayed in the film trying to improve these traits that we all sort of generally right now agree shouldn't be changed, like, height or physical appearance or intelligence or things like that. I'm more thinking about, again, the unknown unknowns. Again, this for me is very personal. I have a daughter who would not exist the way she does if this technology had existed in that form 15 years ago. So I can't let that one go. Like, maybe I would love the kid just as much. And maybe she'd be just as amazing as she is. She probably would, but I wouldn't know that. So that's what I think. I'm just inherently a little more cautious. Not pessimistic, but just maybe cautious.
Well, I think it's a good note to end on. I mean, caution, with ultimately we're dealing with humans here, even if it's the science and the technology. So thank you all. Thank you all for watching and joining us. And Mario, back to you.
Thank you to all of our speakers tonight, and thank you to everyone who tuned in for submitting great questions, both before the event and tonight. It was a really great discussion. As a reminder, tonight's event will be sent out later this week, so expect the recording of tonight's event. And I hope you can join us for one of our upcoming events. We have two great events, one tomorrow night, also at 5:30. We're hosting a panel on the history and impact of racism on medicine and healthcare. You can find a link to register on our website. And then on Tuesday, January 19th, we have the third alumni author conversation with UCSF alumna Diana Hendel, in conversation with Mark Larrett, the President and Chief Executive Officer of UCSF Health. So with that, have a great evening and good night, everyone.